Good afternoon, everybody. We are back for the next session in the broadcasting. My topic will be Meiosis, the chapter of Meiosis. If anybody has a question, please ask your teacher to send it via the iBox. I am Mrs. Netling. So let us start. The question is, why do we talk about chromosomes? Where do we find them? And we find them in the nucleus of every body cell. Now, body cells are often called somatic cells. You need to know the name because sometimes they refer to that in the exam papers. They don't talk of body cells. They use the term somatic cells. And the other word that we use is gametes, which stand for sex cells. And you need to know the difference between somatic cells and gametes. The difference basically will be that the one is diploid. What does diploid mean? It means the somatic cell has two sets of chromosomes. And the gametes will have only one set of chromosomes. And therefore we call them haploid. Now, each organism on Earth has its own specific number of chromosomes, each species. And for humans, we have 46 chromosomes. In other words, 23 pairs. Now, because gametes are haploid and not diploid, they have half a set of chromosomes. They have only 23 chromosomes. The chromosomes are in two sets. You receive one set from your father, and you receive another set from your mother. And every chromosome from your father has a similar chromosome in size and in shape that he can partner off with the chromosomes of the mother. So they are matching one another. And when they match, we call them homologous pairs. You need to know that term. You need to be able to use it in an exam. A homologous pair, that means it's one from the father, one from the mother. They're matching each other. They occur together in the cell. And they pair off in size and shape. They will also have the same genes in the same place. Now, if you are asking what is a gene, you find different genes on a chromosome. Each one forms a segment on the chromosome, on the DNA. DNA is a chromatid, so you have two sets of DNA on a chromosome, and that controls a specific characteristic like the length of how big your nose is, what the color of your eyes will be, and the genes determine that. So a chromosome basically is a linear set or collection then of genes, and every gene for eye color will be at a specific place on the chromosomes. You won't find it once in the one chromosome at the top and another chromosome totally at a different place. They're always in the same place. And that is what determines who you are, how you look. If you look like your dad, if you look like your mother's people, genes are the units of heredity. The place where you find the gene we call the locus. If it's more than one place we're talking of a loci, L-O-C-I. But you need to know that a locus is the position on the chromosome where you find the gene. You need to know the term. If we talk of a complete set, diploid set of chromosomes in the somatic cell or the body cell of the organism, we talk of the karyotype. Now, you've done karyotypes before in more than one of the chapters. I'm just covering it very quickly. The human karyotype, we said we have 46 chromosomes, so we have 23 pairs. And 22 of those pairs are autosomes, which are normal, ordinary chromosomes, and one pair will be gonosomes, the one that determines whether you're female or male. We call them the sex chromosomes, or the X and the Y chromosomes. And in front of you, you can see now a typical karyotype. And if you look carefully, you can see they come in pairs. You can see there they are the same size, same size, same size. And then if I go down here, some of them look a bit funny, but they're still similar in shape and in size. And then if you look here, you can see the one is half the size than the other one. Typical, the female carries all the genes, the male don't carry it so much, he's much smaller. But if you look at this karyotype, I think you all will realize this is a male karyotype. Why? Because the 23rd pair carries a Y and not a double X, which determines that as a male. So you can see that we call them homologous pairs, and it shows if there's any abnormality. If you get 3 chromosomes instead of 2 in a pair, you know it's some kind of syndrome. There are many different kinds of syndromes. They vary in what they cause. So a female basically has 44 chromosomes plus a double X, which gives you 46. And a male will have 44 plus XY. And X is a chromosome and Y is a chromosome, so we get to 46 chromosomes. Then a word that was famous a few years ago, then when they started working on the human genome, we talk of more than 10 years ago, the complete set of genes in an organism's haploid set of chromosomes we call a genome. You need to know the term in case they ask that. We don't do much about it, but you must know that is what we call it. And for every species, you will have the same. Species genomes differ, but a species specific has the same genome. So your mother, your father, they will all have similar genomes. Just a few general facts about chromosomes. You don't get cells without chromosomes. Chromosomes are always present in cells. So cells always carry the hereditary characteristics of a person or an organism. But you can't see them, even by a light microscope, so you can't see them really. You need to have something better to see them. They're very small, and they normally don't appear as chromosomes. They normally appear as long threads of chromatin. It's only when we start with the division that we start seeing the chromosomes. And how that happens is the chromatin will shorten, thicken, become visible as chromosomes, and that is then when we start discussing the process of meiosis. What are the functions of chromosomes? Obviously, they carry your hereditary characteristics because they hold the genes. And then anything that is formed, you've done protein synthesis in the previous session. Anything that comes through protein synthesis needs DNA, so DNA controls the functioning of the cell, and it provides the code for protein synthesis. And those are part of the functions of chromosomes. DNA is part of a chromosome. That's why you can throw this function in here as well. Let's go to the cell division part. We have a cell cycle that occurs, and that is as the cell grows, it replicates, it divides, and that whole process we call a cell cycle. We basically have 3 phases. I think you know them. First of all, the interphase. And you must remember, meiosis do not occur during interphase. We have karyokinesis. That is part of meiosis. That is where the nuclear, the chromosomes are dividing. the nuclear division. Then we have cytokinesis, and that is when the cytoplasm divide and the cell membrane and cell walls grow back, depending if it's a plant cell or if it's an animal cell. So there are these three phases. Karyokinesis is where most of the discussion will be about. That are most of the 4 phases of meiosis, basically part of karyokinesis, and the last phase will include cytokinesis. If I put it in a diagram for you, the diagram, as you can see, go according to the arrow. The biggest part, 3/4 of this diagram, more than 3/4, is interphase. And during interphase, there are a few things that occur. In this case, on the dark pink, I put mitosis. It could have been meiosis, it doesn't matter. But the green part is, the cell normally does during interphase what it's supposed to do. So it's supposed to grow. So a big part of it is the growth phase. And then before we can have meiosis or mitosis, we need to replicate the DNA. You can see that takes some time up. And it must prepare itself for cell division. And then we come into the 4 phases, where the actual division process occurs, the prophase. I think you know all 4, metaphase, anaphase, and telophase. And then cytokinesis. Now don't be confused. Cytokinesis is actually more part of telophase. It is not a 5th phase. It's just to show you that it happens at the end of the process, that I put it in as a 5th phase. But it's not a 5th phase. It happens quite at the end of anaphase, or start of telophase, and it finishes off with the end of telophase. Important thing to know for the exam is the structure of a chromosome. I just want to draw something quickly for you. If I think of chromosomes, we normally draw them like this. But actually there's no specific centromere. This is just a place where they connect. Now the label here is important, but then why I actually draw this for you is to show you. We normally say label this and we say it's a chromatid, or that is a chromatid. But the chromatid is this whole area. This whole thing is a chromatid. A chromatid is not, for instance, if I draw it like this, and I say, what is this chromatid? Okay? But the chromatid is this whole part. You don't have 4 chromatids here, you have 2 chromatids. Okay? And it's amazing how many times they ask this question in exam papers, and it's actually such a stupid question, to label a chromosome. So you need to know it to be able to label it. Let's go back. Okay, there's my structure of my chromosome. You can see my chromatids. And then there, the place where they connect, we call the centromere. Make very sure you know those labels. Because in most of the exam papers that I've looked at, somewhere they throw that in. You must be able to draw it, and you must be able to label it. And remember, whenever they ask you a drawing, you always give a heading. Whether they give a mark for it doesn't matter. You might lose a mark if you don't put a heading in. So remember, headings are very important, whether it's graphs or diagrams, doesn't matter what it is, put a heading in. Rather safe than sorry. Now you learned about replication. So before nuclear division can occur, we need a copy of the DNA. So during nuclear division, the chromosome is a double structure. It has two identical DNA molecules. And we call those two molecules the two chromatids. So each chromatid represents a DNA molecule. Sorry. Just to repeat again what happens during interphase. Why is interphase important? You need to know that. It is the time slot between the different divisions. And that is where the cell does its normal work, what it's supposed to do. And that is also, during interphase, you have replication of the DNA. You must know when replication occurs, it's during interphase. Let's talk about life cycle. You all know about life cycles. And life cycles, basically, why I bring this slide in, it's because you need to know the connection between mitosis and meiosis. Why do I have mitosis and meiosis? The complete life cycle looks like this. I have an adult. The adult cells are diploid. Then, with maturity, we get meiosis, and we form gametes. Male gametes and female gametes. And you can see it's haploid, it has one N. Then when we have fertilization, when those two gametes come together, the male fertilizes the female, we get a zygote. 1N plus 1N give me 2N. So we're back to diploid. And then the baby needs to grow into an adult. So now we go back to mitosis. And that is what mitosis is for. It's for the growth process. And this whole thing we call a life cycle. In your syllabus, they brought back for this current syllabus, they brought back mitosis, a revision of mitosis. You've done mitosis in grade 10 in detail. So I just brought it back into my slides to remind you how it looks, because it is part of the syllabus. They can ask you about it, because in the syllabus they say you need to revise mitosis. Mitosis is not about the sex cells, it's only about the body cells, how the body will get more cells so that it can grow bigger. Now, the other process is meiosis, which we are actually going to discuss. That is the new work, division of sex cells, how gametes are formed. Now, to recap mitosis, it's in the body cells. Remember, other word for body cell is a somatic cell. What is the purpose of it? Growth, first of all. And sometimes when you go with a bicycle and you fall and you break the skin, you need to repair the skin. And that is done also by mitosis. It's for repair. Now, in short, on this side you see a diagram. On this side you see a photo taken through a micrograph. It's a photo through a microscope. You can see there are the chromosomes, but this is not very clear for us to learn from, so they normally give us a diagram. Now, if you look at this thing, sorry, you must know the labels here. Now, you can see the chromosomes there. There's the centromere connecting them. And then you can see this. You must know, although the label isn't there, it's a nuclear membrane. There are my centrioles dividing. That's the cell membrane. We call this first one prophase. What happens here, the chromatin becomes more visible. Chromosomes become visible. And every chromosome, as you can see, consists of 2 chromatids. The next phase is where they gather together on the equator. And you can see all the centromeres on the equator aligned there. And connected with spindle fibers. Another label you must know. They sometimes ask it. That is metaphase. You can identify it here, but it's not so clear. So they arrange on the equatorial plane, and they're connected to the spindle threads or fibers. That is the next phase, prophase, metaphase. This one will be anaphase, where they start separating. The spindle fibers become shorter, the centromere divides, and we have sister chromosomes moving apart. Chromatids, they move to opposite poles. The last phase is telophase, where cytokinesis kicks in, and we're going to form 2 new cells. Chromosomes disappear again, the nuclear membrane will come back, you have a nucleus again, and the chromatin becomes visible. That was the revision on mitosis. We go to meiosis. We also call meiosis reduction division. Meiosis. The big difference is that the number of the chromosomes must be halved. So diploid goes from diploid to a haploid. The reason for that is when I put an X and a Y together, an N and an N, I must get 2. So if I have 23 chromosomes plus 23, I go back to 46, a normal human being. If it doesn't happen, I won't have a normal human being. So this is actually a very important process. And we have 2 here. We have a first meiotic division and we have a second meiotic division, and you must know both. And when they ask questions about this in exams, you need to say it is meiosis, it is prophase 1 or it is prophase 2. Otherwise you won't get all your marks in an exam. You need to determine very specifically which phase they come from. Now, it starts with interphase, where we have the replication of DNA, as we said before. Then the phases stay in the same order than in mitosis. We have prophase, metaphase, anaphase, telophase. With the first phase, we call them prophase 1, metaphase, and all those 1. The big difference here, have homologous pairs. Very, very important. We also call them bivalents. You can see they are drawn here. That is a pair, and that is a pair. Now, what often happens in the exam, they ask you how many chromosomes are here. And then you say 46. I count only 4. So remember, if they give you this picture in exams, you say there are 4 chromosomes. They don't specify what organism this is. This is just a typical diagram of prophase 1. So you count the amount of centromeres you can see there, and then you will know that is the amount of chromosomes. So I can see 4 centromeres, here are 4 chromosomes. Nothing more, nothing less. And that is a bivalent pair. I've colored them, gave them 2 different colors that we can see what happens. Every bivalent gives you split into the 2 chromatids, that you can see the chromatids. Now, what is very important here is you can see here crossing over occurs. In the exams, they ask you, what do you call the process that is represented here? The process is crossing over. If they ask you the label like we do here, the label isn't crossing over. The label is chiasmata. Chiasmata or chiasma. They won't bother in the exam when they mark if you use more than the chiasmata. But the singular is chiasma, and the plural one is chiasmata. But very important, if ever they give you, and they give it regularly, these chromosomes, they will ask you, what is the label there? They will ask you what is A or B, or whatever they number it like, and it is not crossing over. The process is crossing over. You must give the term, chiasmata or chiasma. What happened then, a piece of the one broke off, and it connected with the other one, and there you can see they've swapped a piece of the chromosomes. So in other words, say this was the mother chromosome, this was the father chromosome. Now the mother chromosome has a part of a father's genes, and vice versa. And this only helps to increase the genetic variation. And that is why all the brothers and sisters don't look exactly the same, because this is random. It happens randomly. You can't say before the time which parts are going to break off and connect. And this is part of why we have the variation. And we still have a homologous pair. And we're still busy with prophase 1. We can also say if you get a bracket that includes, in a big drawing, that includes both these, you can say it's ambivalent. That will be the label. Or a homologous pair. Both will be acceptable. We're still busy with prophase 1. You can see it looks typically exactly like the mitosis one, except, okay, this is the beginning. The nuclear membrane is going to disappear now. The nucleolus will disappear. And the spindle is developing. There you can see the spindle developing. There are the centrioles. We go to the next phase, which is metaphase. And with metaphase, instead of, if you look carefully, you can see here we have a pair that's on the equator. If we go back to mitosis, you have a single chromosome on the equator. Here, you have a pair on the equator, and that's the big difference between meiosis 1 and meiosis 2 also. In meiosis 1, if they give you this drawing, they ask you where does it come from, which part of the phase, you will say this is metaphase 1. If they ask you why, it's because we have a homologous pair on the equator. We don't have a single chromosome here. And you can also see that crossing over occurred there. The next phase, anaphase 1, and people, it's very important. It doesn't help you recognize this as anaphase 1 and you only write anaphase in the exam. You have to say anaphase 1, otherwise it goes through like mitosis anaphase, and you won't get your marks. And you can see here what are separating is not a chromatid and a chromatid, it's a chromosome and a chromosome. The centromeres didn't divide. It was two separate chromosomes lying next to one another, and the one goes to this side and the other one goes to this side. So the bivalent is now splitting. And with anaphase, late anaphase, cytokinesis will start. And then you can see here, cytokinesis is continuing. Here it finished in late telophase. But it is still telophase 1. I have a chromosome, I have a chromosome here. And what happened here? We moved from diploid to haploid. Because we had two chromosomes pairs, And we had 4 chromosomes, and now we have only 2. If we will go quickly back, there you can see you had 4 chromosomes in the cell. So pairs, pair and a pair. So this was still diploid. And then when they start separating, we get a new cell forming. And this one will be haploid. And that's still telophase 1. Okay, and at the last bottom here you can see membrane formed everything. We have back a nucleus, a nuclear membrane, only 1 centriole, so cytokinesis complete. We have a gamete. But not a gamete yet, we still need to go another division, which is second meiotic division. I'm not drawing the 2 cells now. I'm only working with the 1 cell here. But remember, on the side, there's actually 2 cells that are dividing here. And now we talk of prophase 2. Prophase 2, because I can see I had crossing over. So if they give you this picture in an exam, and the chances are very good they will use only meiosis pictures instead of mitosis. Mitosis is more revision to see how it works, or how that part works, but this is the more important one for matric. You must be able to say what part of a phase this is. They will color the part black, and the rest will be white, and then you will be able to say this is prophase 2. Because you can see crossing over has occurred. The same story again, spindle fibers, nuclear membrane will disappear. Chromosomes still have their sister chromatids because the chromosome hasn't divided. But now we have this is my second metaphase. I now said that's mitosis, that was second metaphase. This is first metaphase where we had the pairs. And now we have a single chromosome. If it was mitosis, you wouldn't have had a red there. It would have just been the same right through. It's only to indicate crossing over that we know this is, that they color it, and then you know you're busy with meiosis, not mitosis. So you can see we don't have pairs here. It cannot be 1, it must be 2. So we call this metaphase 2. Just to recap, we are in meiosis because you can see crossing over occurred. And we are in metaphase 2 because it is a single chromosome and not a pair of chromosomes on the equator. Spindle fibers are connected, and now with anaphase we find now it is like in mitosis, the centromere divide, the chromatin fibers become shorter, and they pull the sister chromatids to the opposite poles. And that is anaphase 2. And people, they ask this in exam. I went through many papers, and you find it in the papers. And it's actually easy work that doesn't take a lot of understanding. It's more learning. And once you know it, you can get your marks. So they don't ask difficult questions about it. So you can actually ensure that you will get these marks. Late anaphase, cytokinesis will start. And then in telophase it will be completed, and you can see that we have now at this stage, after the second division, we have gametes. Nucleus will be forming again, and the spindle fibers will break down, the cytoplasm divides, that's cytokinesis, and we have new cells. If you remember, originally we started with a cell that's split into 2, so actually we don't have 2 new cells. If I take the complete picture, I will have 4 new cells. I'm talking of 2 because I'm only working with the 1 cell from meiosis 1. So at the end of meiosis 2, you will have 4 haploid cells, and they come from the original diploid cell. And a haploid cell has half the chromosome number. You can't say it's 23 because if I'm not talking of a human, it can be any other number. It's only if they talk of humans that you can say it's 23. And you must remember that. You tend to forget it. And they are not the same because of crossing over. The other thing that I want to show you is when they align here. Let's go back there. When they align here, they talk of a random assortment or random positioning here on the equator. That's part of the variation. Nobody says that all the father chromosomes must be on this side and all the mother chromosomes on this side. They randomly position themselves here. Sorry. So that adds to the variation. Okay, let's go to... Yeah. Now, they can ask you to compare the 2 divisions. They can ask you that. I don't think... Nobody can say they won't ask a comparison between mitosis and meiosis. But I think they might rather ask you between first and second meiotic division. If they do that, you do them in the phases. Otherwise, you may forget some of the stuff. And remember your pictures. You must always, when you learn this, you have to draw the diagrams. You cannot learn this by just reading through it. You have to write, and you have to label. You have to draw the diagrams and tell the story of this phase as you write it down. That is the way you learn this. Otherwise, you're not going to remember it. You're going to forget it. So, to start with, you need to be able to draw them. And from there, you can tell how they work. So when you come to a question like this, you remember the drawing. And then you will remember the story. Now remember in prophase 1, we had pairs of chromosomes. While in prophase 2, they have divided and we have single chromosomes. In prophase 1, crossing over occurred. And in prophase 2, there's no crossing over. So you can see there are many things you can write if they ask you a comparison. If we talk of humans, there are still 46 chromosomes visible, or they are diploid. In the second division, it will be half the chromosome number. So if you don't talk of humans, you can say it's a complete set of chromosomes. And the second division of prophase 2 will be you will have half the set of chromosomes visible. Then I go to metaphase, and remember metaphase is when it goes onto the equatorial plane. So they align there, and because we have pairs, it is a pair that aligns. And in metaphase 2, it's single chromosomes that align with the centromeres on the equatorial plane. In anaphase, we have pairs that separate. Anaphase 1 and anaphase 2, we have chromosomes that split at the centromere and the chromatids move to opposite poles. Then the last phase, the telophase. After telophase 1, you have 2 new cells with half the amount of chromosomes. Telophase 2 will give you 4 new cells with half the original chromosome number. And we call, therefore, this is called reduction division because you halve the chromosomes by number. Now, why is this so important? It's extremely important, as I said when we started, because when you fuse the gametes during fertilization, we want to go back to the original number of chromosomes. And it's important to produce the gametes. Then, which they ask you often later on in some of the other chapters, the genetic variation that is induced with crossing over. and also the random arrangements of chromosomes. As I explained with the mother and the father chromosome, that can be randomly to the left or to the right side on the drawing. If they ask you mitosis or meiosis, I just put the table on. Mitosis is in body cells, meiosis in sex organs. In mitosis you have only one division, meiosis has two nuclear divisions. In mitosis the result, the product, looks exactly the same than the parent, so the chromosome number will be similar and you will get 2 cells from one cell. In meiosis you get 4 cells with half the number of chromosomes. They're genetically different. In mitosis they're the same, and we never have pairs in mitosis, while we do have pairs in meiosis in prophase 1. No crossing over in prophase 1, not 2, remember. So you can't say in meiosis, you have to determine where in meiosis. And then during anaphase 1 we have chromosomes that move across and not chromosomes that split. Okay, now one of the diseases we cover here, but you actually cover it in more detail in genetics, is Down syndrome. So I don't have all the detail of how a Down syndrome child looks. That you will find in genetics. But this is an example of trisomy. What is trisomy? It means that we have 3 chromosomes at 21 instead of only 2. Then there's something else they use these days in the agriculture, polyploidy, and I see they ask it in the exams, so I'm just covering it quickly for you. It's when it has more than a diploid set of chromosomes, we say it's polyploid. And the condition is polyploidy. You get 2 types of them. It can be triploid or it can be tetraploid, 3N or 4N. Now how do I get this? To get a triploid, it means say for instance when the egg cell was formed, that there was a non-disjunction of the chromosomes. In other words, they didn't separate. The word non-disjunction means they didn't separate, so the egg cell stayed diploid. It didn't go haploid. So the one side kept all the chromosomes, and so therefore it's diploid. And now if I add the gamete, which is haploid, I will get a 3N. The next one is the tetraploidy. If the chromosome number doubles during the DNA replication and the cell does not divide, it stays with the 4 sets, or the 4N, and therefore we will call it tetraploid. Let's just look here at the bottom one. When a diploid sperm cell will fuse with a diploid egg cell, it will form a tetraploid zygote. And we use this in agriculture, and we make some of the plant species actually like this because they give us bigger cells, bigger fruit, they look nicer. So in agriculture they create these species. In a lab they use colchicine, and this drug prevents the formation of the spindle, so the chromosomes don't separate, and you produce plants with bigger flowers, fruits, and storage organs. You need to remember where meiosis takes place. In animals, it's in the testes and in the ovaries, and in plants, it is in the sporangia. We tend to forget that we also need to remember the plants. And then just a few terms. Gametogenesis is the all-inclusive term for spermatogenesis and oogenesis. Spermatogenesis is formation of sperm cells. And then obviously it takes place in the testes, while oogenesis is in the ovaries, and it helps to form the egg cells or the orgoniums, and that will be the end of the human. Then we have spore formation, which is in the plants, in the sporangia. You remember the stuff? You think we've done them in grade 11. And that is the end of the slides. If you have any questions, you must please ask me. We're going to break now for 10 minutes, and then we will start with exam papers and questions in exam papers. Thank you.